Hello friends, in this video we will learn about the second part of fundamental unit of life, which covers the topics like structural organization of cells. So let us start with the chapter, and also the last part of the video, that is part 1, is available, whose link is given in the description section. You can check it there also. So structural organization of cell means in this part we will learn about the structures, the different structures which together constitute the cell. Mind my words. In this part we will learn about the structures which together constitute the cell. So here comes the first part or first structure of the cell, that is plasma membrane. Plasma membrane is the outermost covering. Remember, plasma membrane is outermost covering, and it is found in every cell, in every plant cell, animal cell, bacteria, any kind of cell. If it is a cell, then you will get there plasma membrane, which is the outermost covering. It is also selectively permeable. Selectively means it chooses something and it ignores some constituents. Permeable means it allows to pass through it. It permits to pass through it. Remember, plasma membrane, it allows some substances to pass through it. Plasma membrane, it is a plasma membrane. Some substances can pass through it and some cannot. So it is selective in giving permission. That is why it is known as selectively permeable. Okay, we all know that cells need oxygen for breakdown of food and obtaining energy, and after breakdown of the food, that is after respiration, carbon dioxide is generated. So oxygen must pass through membrane and carbon dioxide should come out through the membrane. So gases pass through the plasma membrane by the process diffusion. Now what is diffusion? Diffusion is a process. Diffusion is a process where we can see movement of some substances from the region of higher concentration to the movement of that substance to the region of lower concentration. So this is a region, this is another region. Here the concentration of that particular substance, it may be oxygen, it may be something whose concentration is more, and in this region the concentration is less. The movement will occur from higher to lower, not from lower to higher. Clear? So this process is known as diffusion. So diffusion of oxygen and carbon dioxide occurs through plasma membrane. Okay. Now water also move from higher concentration region to the lower concentration region. So from higher concentration region to the lower concentration region, water moves through the process of osmosis. Remember, osmosis. Okay, now what is the difference between diffusion and osmosis? Diffusion may occur through any membrane or through any space, clear space. But in case of osmosis, water moves through a semi-permeable membrane. From higher to lower concentration of region, there is a semi-permeable membrane in case of osmosis. But in case of diffusion, you will not get any kind of selectively permeable membrane. Clear? Okay. Now here comes the hypertonic, hypotonic and isotonic solution. Okay. To understand this, we need to take a situation like this is a cell. Okay, this is a cell. So inside the cell there is water. We all know that cells have water inside them. Okay, there is a particular concentration of water. Okay, now it is dissolved in a glass of water, So here also, when the cell is inserted in the water concentration is maximum in the outside the environment. Are you getting the point? The water concentration is maximum, is more outside the cell, whereas water concentration is less inside the cell. So what will happen? Water will move from higher concentration region to the lower concentration region. As a result, the cell will swell up. The cell will swell up. Clear? Because water is entering into the cell. So this kind of solution, the solution which is present outside the cell, this kind of solution is known as hypertonic solution, where the water concentration is more. Similarly, when the water concentration is less in the solution, it is a cell, the water concentration is less in the outside solution, this type of solution is known as hypotonic solution. So in this case, the water concentration is more inside the cell, water concentration is less outside the cell, so water will move from inside to the outside of the cell. As a result, the cell will shrink because water is coming out of the cell. Here water was entering into the cell, here water is moving out of the cell, so cell will shrink. But in case of isotonic solution means the concentration of water inside the cell and outside the cell is same. The concentration of water outside and inside the cell is same. In that case, there will be no movement of water because the concentration difference is not there. I think this is clear. Okay, next part we have cell wall. Okay, we all know that cell wall is found only in case of plant cells. In animal cells, we do not observe cell wall. In case of plant cells, we have cell wall. Now, plant cells have plasma membrane as outermost covering. Okay. Additional to this covering, there is another layer known as cell wall in case of plant cells. Cell wall is very tough, very rigid. It is made of cellulose. Next, we have nucleus, which is known as the controlling center of the cell. So this is nucleus and it controls the whole cell. Remember, this nucleus controls the whole cell. Okay. Without the nucleus, the cell cannot exist. Nucleus is double layered cover. Okay. Nucleus is double layered cover. If I draw a particular nucleus, the nucleus will be like this. It is double layered structure. So this is a nucleus. I am not drawing the whole cell. I am drawing the nucleus here. So we can see here two layers. Two layers. 1, 2, 1, 2. Two layers. So double layered cover. And they have pores. We can see here some pores. Through these pores, substances can move in to the nucleus or can move out of the nucleus. Clear? Next, we know that nucleus has chromosomes. Nucleus has chromosomes. And we also know another term known as chromatin. So we should know the difference between chromatin and chromosome. Actually, when a cell is at rest, remember the cell may be in two phases. Either it is at rest or it is dividing or about to divide. Clear? When the cell is at rest, the genetic material which is present inside the nucleus is known as chromatin. It is simply just like thread, a coil of thread, chromatin. But when the cell is going to divide, when the cell is about to divide, these thread-like structures forms rod-shaped structures like this. These structures are known as chromosomes. So chromatin and chromosomes are basically same, but their structure is different, their shapes are different. And chromatin is found in the resting cell, chromosomes are found in a dividing cell. Okay. Chromosomes has DNA which possesses the information and also has protein. That's why chromosomes, DNA and protein known as chromatin, which is found in the resting stage. Okay. Now in case of prokaryotes, they do not have nuclear membrane. Remember, prokaryotes do not have the nuclear membrane. Otherwise, they have the DNA and protein, but they do not have the nuclear membrane. It is not like that prokaryotes do not have nucleus. They have nucleus, but they do not have the nuclear membrane. Whereas eukaryotes have the nuclear membrane along with its constituents. Clear? Now we will move to the cytoplasm. Okay. Cytoplasm. The concept of cytoplasm was already given in the last part of this chapter, Fundamental Unit of Life. In the description part, you can get the link to the part 1. Okay. In cytoplasm, we find some structures. Remember, in case of cytoplasm, we find some structures. Cytoplasm is the matrix or liquid part which is found outside the nucleus. Remember, outside the nucleus known as cytoplasm, not inside the nucleus. Inside the nucleus, it is no more cytoplasm, it is nucleoplasm. So outside the nucleus, it is cytoplasm. So they have some cell structures known as cell organelles. Okay. There are many cell organelles in cytoplasm. In a cell, some cell organelles has membrane, means some cell organelles has covering, and some cell organelles do not have covering or do not have membrane. Okay. In case of prokaryotes, if the cell is prokaryotic cell, naturally they do not have the nuclear membrane and they do have the cell organelles but without membrane. Those cell organelles which are having membrane, those cell organelles cannot be found in prokaryotes. Only cell organelles which do not have membranes, those cell organelles can be found in prokaryotes. But in case of eukaryotes, this is very simple. Any kind of cell organelles, whether membranous or non-membranous, can be found there, and the nucleus is having the double layered nuclear membrane in case of eukaryotes. Okay. Now next cell organelle is ER, endoplasmic reticulum, which is found in the cytoplasm. Okay. Endoplasmic reticulum, it is also having membrane bound tubes, means they have membrane. That means ER is not found in prokaryotes, it is found in eukaryotes. Okay. These are membrane bound tubes. Like inside the cytoplasm, you will get some this type of structures. You will get this type of structures, tube-like structures connected. So these are known as endoplasmic reticulum. ER are of two categories, SER and RER, smooth endoplasmic reticulum, rough endoplasmic reticulum. It is rough because the surface of ER, in case of RER, the surface of ER is having some ribosomes. So due to the presence of ribosome, the surface becomes rough. That is why these ERs are known as rough endoplasmic reticulum. But some ERs are there whose surface is not having the ribosome. So in that case, those ERs are known as smooth endoplasmic reticulum. So very simple. Those having ribosomes are rough. Those do not having ribosome, those do not have ribosomes are known as smooth. Okay. Ribosomes are also cell organelles. These cell organelles helps in protein synthesis. These are site, the place for protein synthesis. So RER, rough endoplasmic reticulum, they are having ribosome. So that means they help in protein formation. RER helps in protein formation due to the presence of ribosome, whereas SER helps in lipid formation. And both lipid and protein are found in the plasma membrane, cell membrane. So these lipid and protein synthesized by smooth endoplasmic reticulum and rough endoplasmic reticulum are used in the formation or synthesis of plasma membrane. This phenomenon is known as membrane biogenesis. Genesis means generation, formation, membrane means plasma membrane, bio means living. Okay. Now in liver cells, we find also, because it is cell, so we naturally find smooth endoplasmic reticulum. This smooth endoplasmic reticulum performs detoxification. Now detoxification means whenever your body is having any kind of poisons or toxins, poisonous substance or toxins, your liver clears these substances from your body. It helps in elimination of these poisonous or harmful chemicals from your body. That's why if a person consumes daily alcohol, his liver is the first organ which gets damaged, because liver helps in detoxification due to the presence of SCR. In this video, this much is enough. In the next video, we will be coming up with the next cell organelles in part 3. Thank you very much.